Hello, review committee members. My name is Amita Kaundal. I am an assistant professor at Department of Plant Science and Plant Soils and Climate in College of Agriculture and Applied Science at Utah State University. I am presenting my proposal entitled Microbial Diversity Analysis of Dwarf and Common Bunt in Different Soil Composition to Study Disease Suppressive Soil and its Microbiome. With increasing bird population in this era of climate change, the demand for food production will increase. We need to develop new methods to meet this demand. Wheat is the second most grown crop worldwide. Dwarf and common bunts are the diseases of wheat caused by soil-borne fungal pathogens from Tilletia genus and cause significant yield and quality loss. There are four main approaches to control these diseases: use or development of resistant genotypes, seed treatment with fungicides, use of clean seeds, and early planting to avoid the disease in case of dwarf bunt. Both diseases are soil-borne, where the soil gets contaminated by the spores. The soil microbiome is abundant in plant growth promoting bacteria, which can help suppress diseases. Therefore, disease suppressive soil can serve as an alternative method for controlling these diseases. The unique microbial communities in these soils compete with pathogens for resources and produce antimicrobial compounds. During a screening of wheat resistant lines for common bunt in Dr. Chen's lab in University of Idaho, they observed that the soil from the field used in the greenhouse pot studies suppressed the progression of common bunt compared to the soilless media. This suggests that the field soils has disease suppressive abilities. Therefore, in this proposal, we want to investigate the disease suppressive microbiome of the field soil from Idaho, field soil from Logan, and soilless media mix with resistant and susceptible genotypes infected with dwarf bunt and common bunt, and one set without infection as control with two objectives. So in first objective, we would like to study the effect of field soil and soilless media on the progression of common bunt and dwarf bunt on resistant and susceptible genotypes of wheat for both diseases. For the second objective, we will conduct the comprehensive metagenomic analysis of bulk, rhizosphere, and root microbiome to identify core microbiome and keystone species. We will keep 5 pots for treatment, a controlled dwarf bunt and common bunt, in the plots outside in the field, as dwarf bunt need snow cover to induce diseases. One set of the pots with common bunt and control we will keep in the greenhouse to establish common bunt diseases. We will collect disease incidence data for objective 1. For objective 2, we will carry out a comparative microbial diversity analysis, core microbiome identification, differential abundance analysis, and a microbial co-occurrence network will be created between resistant and susceptible genotypes in different soils with and without pathogens. These analyses will help us identify the core microbiome and keystone species. It will give us preliminary data to get funding to isolate beneficial microbes from the disease suppressive soil, test them for biocontrol activities, identify the mechanism involved in biocontrol activities, and develop a potential core microbial resource bank to construct a synthetic microbial community, SYNCOM, to control dwarf and common bunt. These syncoms will be used as a biocontrol agent either to treat seeds or directly amend the soil to control dwarf and common bunt infections. I am collaborating with Dr. Felipe Sabadin at Utah State University and Dr. Jinli Chen at University of Idaho, who will help me selecting genotypes, provide inoculum, and score diseases. Both of these faculties has experience working with wheat and these pathogens. Thank you so much.